welcome you all. So here we'll be discussing a question related to antigen antibody interaction. So this question is from GATE 2014 Life Sciences question paper. So this is from GATE 2014 Life Sciences question paper, biochemistry portion. This is question number 6 and the question is, the affinity of an antibody can be determined quantitatively by which of the techniques? So 4 techniques are given and we have to find out the affinity. So what is an antibody? So antibody is a molecule that can react with an antigen. Okay, so that antigen also in the antigen, the part where the antibody is raised against, that is called as epitope. So what we do in our experiments, we need the antigen antibody interaction readout as many of our experiment data. So what we do, we give a certain antigen, and that antigen we inject in a particular animal or something, allow that antigen to go through the immune system processing of the animal, and then after few days that animal raises antibody against that antigen, and that antibody we can collect in the form of sera. So that sera now contains the antibody against the provided antigen. So what we need to do, we need to check the affinity of that antibody and the antigen. So antigen is nothing but the protein molecules that we have, and in the sera we have the antibodies. Now if you want to check the interaction, there are several techniques. Okay, you can check it by Western blotting. That is a very ready to do techniques. Other than this, if I'll see from these options, so the first option is the Malditov MS. MS is nothing, is it a mass spectrometry technique, where this particular technique is used for protein quantification, protein structure prediction, protein sequence prediction mainly. So what are the sequence and everything. So you uh, isolate proteins and subject it to mass spectro. There you can recognize the sequence of the protein, as well as you can recognize the interacting partners of the proteins, and those are the things that can be done using the mass spectrometry. And that for mass spectrometry, you can pull down the protein using the antibody, but that doesn't tells you about the quantitative affinity, the affinity in the quantitative manner. Okay, so the Malditov MS is not specifically used for the quantitative analysis of interaction of antibody and antigen. Okay, second is the isoelectric focusing. Isoelectric focusing is nothing but, so it is a technique used to find out the isoelectric point of many proteins. So if you have many proteins in a different tube, you have many proteins, and then you set up a pH gradient from one end to another end, and then you run the current. So protein, according to their charge, move in a separate direction. If negatively charged protein, it will move towards the positive charge. Positive charge, it will move towards the negative charge, and until it reaches a certain pH. So here also we have a pH gradient. So when it reaches a certain pH where the positive and negative charge are equal, okay, that is the isoelectric point. So that can be determined using this isoelectric focusing technique. So the protein gets aggregated at certain point, and that tells about their pi. So here if one protein is there, so at 2 it is aggregated, that is means its isoelectric pH is 2. If this one, suppose it is at 7, so its isoelectric pH is 7. So isoelectric focusing has nothing to do with the affinity of antibody study. So it is a technique used to tell the isoelectric point of certain proteins. Second is the STS page. STS page is sodium todecyl polyacromate gel electrophoresis. In gel electrophoresis, what we do, we run the protein sample, and the protein sample gets separated according to their size. Along with that, we can run a marker to identify the size of a protein. So page we perform for the separation of protein molecules on the basis of their sizes. So that has also has nothing involved related to antibody. If we see the fourth technique, that is the equilibrium dialysis. So in equilibrium dialysis, what is happening? So equilibrium dialysis is dialysis technique. What happens is you equilibrate something. So suppose if I have a beaker divided into two chambers, okay, and it is filled with buffer. So suppose I perform dialysis here. So here in this is A section, this is B section. So here, if I suppose I have added 6 protein molecules and allow the dialysis to take place. So after the dialysis, what happens? It's try to equilibrate, okay, because of the equal volume and everything. The protein molecules will be equally separated because this is a porous layer that can be crossed by the protein molecule. So at the end, proteins are okay. This is how they are getting equilibrated in section A and B. So if you take out these both A section and B section, and then you study, you will find 3 molecules of protein here and 3 molecules of protein here. That is, it is equally distributed in both of the sections. However, if in this particular setup only, what can be done is you make a setup, again give so in the same setup, section A and B. So in section, so this is a quantitative method. Okay, so suppose quantitatively we are giving 6 molecules of protein, and there will be practically it will be different. Some concentration, particular concentration will be given. So here in a practical way, I'll be showing that if 6 molecules are given, and here if suppose 2 antibody molecule I am giving here. Okay, so after the dialysis, what we will see that, so this 2 antibody will make complex with 2 antigen molecules, and now we are left, so remaining antigen will be equilibrated. So now if you take both the proteins, so if you see detect for the free antigen, you will see 2 and 2. There also the number is equilibrated. For example, in our previous case where 3 and 3, even though there that is the control where we were getting more because we have given 6 molecules, so 3 and 3 distributed. Here also what we are getting total 4 molecules. That means the remaining 2 molecules of protein are involved in interaction with the antibody. So that is how you can detect the affinity of antibody to an antigen. So how many antigen it is attract, that to on a quantitative basis. So that is what a equilibrium dialysis. Using the equilibrium dialysis, you can identify the affinity of antibody quantitatively towards the antigen. So the correct option out of these 4 techniques is the fourth one, that is equilibrium dialysis. And this is my NPTEL reference. You can read more about these antigen antibody interaction and here. Thank you.